audible?
Yes, how are you? Hi.
Yeah. Hello, hi, sir. So I'm fine.
Good.
So today,
Okay.
so should I start giving my brief?
Okay.
Yeah. So basically, let me share my screen first.
All right. Hello, Mayukh.
Basically, as for the last time, I have been suggested I have to make a color changes. So as you can see, these were the light gray color. Last time I have made them black as per the opinion. So this is one task that I have completed. Another task that I have done is I am seeing how to do more better and better segmentation of CWs and Android. And I have come across a paper. This is the 3D MMS, robust 3D membrane morphological segmentation of CWs and Android. So these are in this paper they have given they have taken very strong neural networks to segment these C.elegant membrane basically. And I will give if next week allows I will like to give a presentation on it, a short presentation.
Okay.
So basically so if I show you the results, so this is basically the result. So this is the C dot so this is the C dot element membrane, and these are the partitions that segmentation that we are trying to achieve. So they have done this in the real time. So these are the ground truth table that they have made, and these are the results of. So they have named this model as 3D MMS. So if you let me share the link of this paper.
Okay.
So they have also made their code public called 3D MMS module. So I was looking through it. So it is basically based on MATLAB completely, but we can use Octave libraries to import this code into ours. So if you can see, let me zoom it a bit. So if you can see, they have made a GIF in which they are doing this kind of segmentation in real time. So this model is basically based on a MATLAB model, which is basically based on TensorFlow and PyTorch. So they have I'm still learning about how they have made this model. But the initial intuitive idea is they have used the same mask technique, they have created the ground truth tables, and they are just implying masks over it so that they can get more and more good results. And they have annotated a lot of images. So in the coming week, in this week, I am going to study this paper thoroughly and try to see how they are doing these things, because the segmentation they are doing is really interesting, and if we can achieve something like that, it will be great.
Yeah.
Apart from this, I have also started making the image editor for the DevoWorm portal. So basically, this is I have two options, whether to make a complete React app using React UI and or just save your basics on JS. So we can achieve almost all the things that we need, or we don't need a very super advanced type of photo editor like Photoshop or something. So we can save through basics. I have created this thing in JS. So as you can see, you can upload it from a computer or create a new image itself or sample it or use a sample. So this is the sample image that I have used. So it has some options like cropping the image, rotate image, so you can rotate the image on a particular angle itself or just directly this. You can resize the images. This has some problems right now, like after rotating the image is not visible. So this type of things I will definitely improve when I submit it. So you can resize this according to your considerations. Also, like I think someone wants to just get an online tool to annotate and label their data. So you can just go up and like this, you can annotate, hand label your data mask, and I will working on exporting these coordinates in XML so that they can get graph tables really fast. So this is not completely working right now. For example, the filters are not working right now, but I will, as we proceed, I will make sure that all these things are working.
Yeah.
So this is the update from my side.
So this is something you created, the editor?
Yeah.
So is this something you built from scratch, or did you build like?
Basically, like there are JavaScript libraries which let you, like for example, like for the filters, there are open source libraries which let you import the JavaScript in your original script, and then you can just use them. So like the noise filters, gradient filters. I hope everyone has heard about them. Like the noise and gradient filters are very famous, like multiply filters. So I'm trying to step it out, like in the editor.js, what kind of features we can use for our purpose, and just trying to fit it in.
That's pretty impressive. Yeah. Thanks for the update. Let's see. So you said the first thing you had was a tab where you had the DevoZoo. So
basically the text is in gray at first, so now it is in black, as suggested.
Right. Thank you. Yeah, that looks better. I can see the text now, so yeah.
The second is like paper, which I have shown for T.elegant segmentation. So like it is a really interesting approach that they have taken. Like they are trying to model the T.elegant embryo at the same time doing the segmentation. So this is something we can definitely look into.
Yeah.
So this is their source code that they have provided for the public use. So we can see like if we can get something from it, it will be really great, I guess. And then there comes this image editor. It is not completed yet, but yeah, it will be completed by then.
Oh, that's great.
Can you hear me, Ujjwal?
No, yeah, it says.
Yeah, is there a way that is there a way where we can run that website locally in our machines from the repo?
Currently, like I have built many parts. It is segment, like it is not actually integrated. These are different parts.
Like working on the website, like DevoWorm.ai.
Like So like
we run it on our machines, like in a way, just to get a better look, because we just look at it once a week. So I think better feedback when he looks at it himself on his screen, right? So like give us some instructions, like on the README maybe, on how to run it on our machine so that we can just.
So basically, this is what I'm trying to say is like these are different components that are not connected. For example, like for the image editor thing that I have built, I'm pushing it. So you can go to repo, you can see the Devo editor name tool, and you can just click on the photo editor HTML and it will start working. But it is not right now.
A small guide on the README so that we can just run and check for feedback, you know.
Yeah, so I will definitely prepare a whole documentation like how to install and run it on their own. But the documentation makes sense when all the parts are connected together and it is working as one. Right now, these are separate parts that I'm building, and after a while, I will merge all these parts together, and then it will become a perfect portal. So just you can type like one command and it will start running.
Yeah, fine.
So yeah.
Yeah. Well, we might want to revisit this maybe once or twice before you implement it too, just to make sure that it's working before, because like if you try to implement it all at once, I would say that it's a little bit, maybe it might be a little bit tricky to get like to get it working correctly, and then people will have comments. So I don't know if, like, I know you can't really implement it on its own and have people test it out, but it would be nice to have like a way for people to evaluate it before implementation.
So I will like in the coming weeks I will definitely integrate all the parts together. But like point is, if you have integrated, like you are building up one application, and if something go wrong, so you don't figure out like what is happening, like what is wrong. You have to figure it out, and you have to delve into code deeper and deeper to see like what is wrong with this thing. But if you are making it in a small parts first, and then you are taking bottom up approach, like you are creating all the separate parts and then you are combining it together once these all are completed. So this becomes easy, like like if you have recently added this part is perfectly working, but when you added it into your main portal, it's somehow not working. So like something is wrong in the integration of this part only. So that's what I'm thinking while I'm building this.
Okay. Yeah, that's good. I think that's very impressive. Thank you, Ujjwal. And Vinay's here. He missed this part, but it'll be okay because it'll be on YouTube. You can watch it. Welcome, Vinay. Now, Mayukh, could you give us an update?
Can you see my screen?
Yeah, it's coming up.
So I've been working recently on that. I went back to the Epic dataset. Like I did some stuff on it during my phase 1. So this time it was more about integrating the video data with the annotations. So the first problem that I was facing was that, like as you can see, wait, I'll show you. Can you see it? Like, is it zoomed in enough? Like, is the text large enough?
You can read it, yeah.
So the problem was that the image, like the number of image time points was 237.
Yeah.
Okay. And the number of edited time points was 135. That is fine. But the number of frames in the video was 237 only. So the first problem was that I had to redistribute those 237 frames into those 135 time points to do something with it, to do anything, to at least like to at least map the frames with the data points within the metadata, like those CSV files. Okay. So that was great. I'll show you. I think it was this one. Yeah. So initially I just had to do that one, and then I had to reformat the data completely, and completely, because like we had to distribute it through 200, we had to distribute it uniformly through 200 frames, 200 plus frames. So after that I started plotting, like categorically plotting the what do they call this thing, like the populations of each cell lineage from during the embryogenesis until the L1 stage. So as we can see, like they basically double after every n minutes, most of the families actually. The others are not very visible because of the scale of the chart. Otherwise it was kind of visible when I just viewed them individually. They are doubling. So I thought, why not make a model which would look at a picture, like a frame of the embryo, like the ones given on the Epic dataset, which is pretty huge, and just estimate the population of each lineage of cell. Like we'll be estimating the population of A, E, M, P, C, and all of these sub lineages from just a frame. So I had to reformat the data and all that. So I'll just skip these parts, which are like the ugly parts basically. So then I just looked at the frames, and what I saw was that they were very noisy, and they were too noisy to actually do something with them. So I had to first remove the noise, and like after doing some basic stuff with OpenCV, I could just smoothen out the images a little bit so that there's no noise but the data is retained. You can see the samples right here. Can you see it?
Yeah, I can see it.
Yeah, so these are like the denoise samples. So I've retained almost all the data by denoising them. So after that, I moved on to like I just trained a deep learning model on it. And what it does is that I'll just keep these parts. Like you can see the data is like as follows, like this picture, and the label is like 3204, and all of these values, they are the populations of each cell lineage at that given for that given photo. Okay, so you can see in other like different photos there are different populations, and most of the times what I've noticed for specifically is that they are very like most of them stay in powers of 2 because they're doubling. then I realized after looking at the videos that they're doubling, which is why they are mostly in any power of 2, like 32, 64, 256, 2. Yeah, so then I'll just keep these parts, like then I trained a model. And for now, the results are not very great because I trained it on very little data. Like little data, I'll show you. Like there are so many cells, like there are so many embryos, and I only had the time to like do it for 2. But now my next objective would be to automate the process to download all of this data by just web scraping, and then like I would make it efficient enough so that it will automatically download all of the data from this website and then format it and do whatever is required and prepare the data so that I can train on it. For now, it's only trained on the first 2, I guess. So after training on the first 2, like these are the kinds of results I'm getting, which are like not too bad, but still they're not the best. Like 64 and my predictions like label and Fred. You can see it, right?
Yeah.
I'll just zoom it. Can you see it better? Like, is it better now?
Yeah, it's pretty good actually.
Yeah, the predictions are like close, but they're not the best. For now, these are good enough. Like, I think this will get better with more data. There's a lot more data. I only use 2 out of 120, I think. So yeah, that's pretty much it.
So this set of images is what you've got. So you've got a result from your model, and this is what it is, like the result.
Yeah, and these images, they are from the the model did not see these images before. It's not like these. The model did not train on these exact images before. This is from the validation loader.
Okay.
So the model never saw these images. This is the first time it's seeing these images.
Okay.
I trained it on the train loader, and this is a small slice of the train loader which I kept separately, which the model would never see. That would only be used for testing. So this is from that slice. So this is like completely unknown images to the model, and these are like I didn't augment them yet because I didn't find the need to augment them. I just flipped and did some stuff. I had to augment them, but my prime objective for now is like just to expand and like scale it up for all of this data. Like there's a lot of data here. So I had a question actually. That the question was that like all of the images that you see in this case, like these images, they are only from one strain. That was the CEH32 strain. Yeah. So is it a problem if I use all the like? Are there certain differences if I use all the different strains? Because there are a lot of strains here. So would it be a problem if I use all of the strains? Because is it different for different strains?
I think well, it's not clear. I guess they're in this data set. They are using all basically wild.
Like there are so many strains. Like
yeah,
like genes. I guess yeah, genes.
Yeah.
Like.
So what that means is that they're expressing different. So what the strains are basically, I think in this data set, is they're expressing different genes. And so you see, if you go back to the images for the different strains,
yeah,
you can see the green dots on the right there. Those are like,
which
well, in the movies, they're fluctuating, but there are slight differences in like the intensities.
Like these dots, right?
Yeah,
these dots.
So there are slight differences in the intensities. So it's expressing different genes in these cells. And so sometimes the cells will be...
I can fix those things, like those are small things. Like I can fix those intensities by just randomizing the intensity on each image. So the model would learn to ignore the intensity.
Yeah.
So that is possible. But I was asking whether or not is it a good move to go for all the strains? Like would that somehow affect? Like it would be like putting dogs and cats into the same category. Would like?
Well, no.
I don't think that.
The only difference you would have in the embryo in terms of cells would be just like there's some mutants that have like don't have certain cells, but those are I think they're all dealing with wild type genotypes. So the strains are just like basically a line that they raise, and so the strain is, it's not really that much different in terms of the cells that are born and that survive. So you're going to get the same cells in roughly the same position. It's probably no more different than like any sort of stochastic sampling error you might get with the cells and taking the image of the embryo. The only thing that would be a problem would be the intensities, but you can normalize the intensities
Intensity is that I'm already actually normalizing all the images. Like, wait, let me show you then. It's there, I guess. Yeah, I'm already normalizing them between -1 to 1. Right. So that's not a problem. Okay. So that's not much of an issue. And I'm already doing some small transformations, like a random rotation, like only like of 10 degrees, and those kind of things. Yeah. And just some basic stuff like vertical flip, horizontal flip. Nothing very fancy right now, and some random brightness contrast, like to counter that thing which you just said. Like there are different brightness and contrast levels. So for that, I'm just randomizing it for every photo. Okay. Yeah. So that's pretty much it.
That's good.
Yeah.
And so this part is for the data.
This part is where I actually load the data. Okay. Like this is a custom class which I had to build. Like first, what I did, there was a very unique problem which I kind of faced. It was that like the population for. I'll just show you the chart. Like you can see this, right?
Yeah.
Yeah. What was happening is that the population of A is way larger than all the others. So what the model was doing was that the model was only approximating the population of A very well, and it was doing very bad with the other populations. Because the model is seeing that it can go to a very good minima just by identifying A and not the others.
Yeah.
Okay. So that was kind of, I was seeing that it was doing A almost perfectly, but not the others. But then to fix that, what I had to do was that I had to clamp all of those values down to 0 to 1. Like as you can see already even on the first 5 columns of the data set, like A is already hitting 4 while all of them are still at 0. So A has a pretty large value and it affects the loss much more than all the other values. A difference in A is a lot costlier to the model than a difference in Z or D. So then I had to clamp all of them to between 0 and 1 so that all of them matter equally to the model. So then now it's performing better, but not the best. I have to scale up the data. Yeah. Right. So that's pretty much it.
Well, yeah, I like that solution because so if you go back to that graph, the one with the cell.
Okay. The population. Yeah, this one.
Yeah, that one. Up.
This one.
Yeah, that one.
Yeah.
Yeah.
Okay. So yeah. So you showed me this on Slack, and so a couple of comments. So these letters are for sublineages. And A is the most abundant in the embryo. So that's roughly half of the embryo is AB, the AB lineage. Yeah. And then the rest of these are kind of like in the second half of the worm, but since there's so many of them, you get like a very small number of them total to work with. Z is by far the smallest because it's part of the germline. Now, that's good that you were able to normalize that because it's there are probably going to be some irregular trends in these other sublineages because of just the way they divide and what they do. But I think A is probably good to see that it actually works to predict the sort of doubling rate. Yeah, I think that's a good graph, and I think you did a pretty good job on normalizing the data.
And I had another thing to say actually. It's that I think it's about time that we start kind of planning about integrating these kinds of models into the website.
Yeah, I think
because there are 2 models by now, so I think we should start talking about that. Maybe next week we should start about this.
Okay, Ujjwal?
Yeah. Yes, like we can
time because we have to first decide which models to take for now.
Yeah.
Yeah.
So what I think is
maybe.
What I think is like we can definitely start on the models that we have completed. So we have 2 things to do with the models. First, we have to integrate on the website, and second, we also have to export it as a Python library so that any researcher can use it just by importing it in a Colab or Jupyter notebook. So we can definitely start from the next week, I guess.
Yeah, we should start doing. I was actually in my timeline. I wrote that I'll do the Python library part on phase 3. Phase 2 was just to polish the models and make them better, and if possible, make more models.
Yeah, okay, sure. Like you can definitely like work on the model.
I'll be done with the Python library by the end of phase 3. But I think what might take time for me and for you is to integrate those models into the website and to see them like come to life. because you have to test them as well before deploying it and moving it to a public domain, right?
Yeah, so we can like do it from the next week, I guess. I have no problem with it.
Yeah, maybe after 1 or 2 weeks because I need some time to
Yeah,
finish things.
Yeah.
sure.
Yeah,
Yeah, so I mean, I would probably, keep me in the loop on this, but I would coordinate between the two of you on that, how to integrate it, because Ujjwal, of course, is going to be doing the integrating, some of the integrating, and Mayukh has to refine the models. And then Ujjwal, of course, has some models that he wants to integrate as well. I mean, I don't know how you're doing it, Ujjwal, but yeah, it's something we should probably
Like,
talk about more.
yes, so I guess like from next week, I guess we can dedicate some meeting on this, like how we want to integrate and how we want to make things work.
Yeah.
If it is fine, Mike.
yeah, we can discuss next week.
Yeah, so we can do that. We can start
We
working
can,
on
we
that.
should, we should do, we should put it on the projects, I guess, on the
Yeah,
GitHub
yeah.
repo
Okay.
for next week or the week after maybe. Like start integrating
Okay.
because there needs to be a like we have to build a bridge, right?
Yeah.
We have to
Yeah.
build a bridge.
Yeah.
There
Does
are two
anyone
sides.
else? Does anyone else have any questions about the updates before we move on? Vinay or Jesse?
Like with Mayukh's work, like this way you're planning to improve these models. Like what are you trying to improve? Like where do you see improvements in this?
Yeah, improvement, like I'll improve by actually scaling
Like
up. Like currently I'm training on very little data. Currently I'm training on like, wait, I'll show you. You can just multiply, wait, 32 by 13 is like, it's a pretty small number, right? 32 multiplied by 13 is like I think it's around 400 frames. I'm training on only 400 images right now. That is only two of all of these. Like I
yeah,
took
okay.
the first two of all of these. So that is only 400 frames, which is
Yeah,
not
yeah.
enough. That is not enough for a good model. Okay. So I have to automate the process of downloading all of these movies, converting them to frames, reformatting those CSV files, and then bringing all of them to a proper format for deep learning, and then train a big model which can hopefully do better. Yeah, so that's some
Oh,
improvement.
have you like tried bulk access? Like I don't know, that is showing some zip folder. Maybe it's a folder for all the data.
Yeah, bulk access is for all the CSV files. I checked. Bulk access is for
Oh,
all the CSV files. So I'll just download the CSV files from this. But I have to automate the downloading of movies. Like I can't just go on clicking and downloading and arranging,
yeah,
right?
yeah,
That would be a mess. So yeah, I'll just download the CSVs from here. I've already downloaded. In fact, I've used this bulk access file before in some other, like in my repo, there is another small deep learning model which does something else. It classifies the lineage from the positions and size and the time value. But that's a different story and a different model. And in our case, we need the images. That was only from the metadata, and this is integrating the metadata, like building a bridge. That model is the bridge between the metadata and the movies. Yeah, so that's it. That's what I'll be doing.
Yeah. That's good.
Yeah, so
Question.
Yeah.
Hope that answered the question.
yeah, sure. Like I'm seeing the website and I see the bulk access. That's why
But they didn't give the bulk access for movies. I don't know why.
oh,
That's the
okay.
problem. Yeah.
Okay. Well, thank you for the updates. We'll go. Let's see. Let me share my screen now. Yeah. Thank you. Present now. I wanted to cover a couple. Well, the rest of the meeting we'll cover a couple things, but the first thing I wanted to talk about was the. Well, we'll put those issues up on the board. So let's go to the. You can see my screen here. I'll go to GSoC 2020 and we'll go to the project board. projects and tasks for GSoC students, and we'll put this issue of so we're integrating the models. I guess that's a good way to put it. And then that's, for now, that's good. And then we can subdivide this up into different issues. We'll put that on hold for now. And then we'll actually assign it. Let's see if I can assign it to Ujjwal and Mayukh. There we go. And then I'll put a DevoWorm in there as well, so we have it. And then we'll revisit this at a couple at meetings in the next couple weeks, and we'll kind of get a status update and talk about ideas if people have ideas about how to do this. And we can talk about it then. So we have a couple. Let's go through this board. We have it up. So integrate cell tracking with segmentation. Where would you say that you are on that, Mayukh?
Yeah, I kind of put it on hold after phase 1 started, so I'll just start it out after I'm completed. That's my second priority right now.
Okay.
First thing is integrating the models with the website. That's what I'll do.
Good. Are there any other tasks that you wanted to add to the board, Mayukh, in terms of like for some of the stuff you're doing now?
No, not exactly.
Okay.
I was thinking of making a movement prediction model, but would that be useful for the worms? Like, it's related to the work that I did before phase 1. Like, remember, I did some stuff which I just made a light version of the tracker to track worms.
Yeah.
So I was thinking, why not make a model that can predict the movement, like the direction and so the future coordinates of the worm. Like, would that be a useful thing for DevoWorm? I was wondering.
I think it would be. I think it would be because we're kind of interested in sort of maybe like behavior that's happening in the embryo, the movement, and all that. So I think that would be a good way to sort of summarize a lot of those data. Where the cells move, and there's some like movement there's a lot of movement in the embryo actually. So it's
not the embryo. Like in the embryo, I can do, it's possible.
Yeah.
But it's a little hard because it's not like a very predictable thing.
Right. Yeah.
And it is not.
As far as I saw.
And it does not even have like much movement, I guess. Like in the worm,
there's no trend.
Yeah, exactly.
If there are trends, then you can train an LSTM model to predict that trend. That is tracking the movement of the worms instead of the cells within the embryo.
Okay. But no, I think it would be more generally useful too. I mean, you could, but I mean, I might do that like later on when you're done with some of the stuff you're doing right now.
I'll set it to the side for now.
Okay.
Yeah, so for now, I think after training this model, the one that I'm currently training, after training this one, I think we should start integration.
Okay.
If anyone has any rough model ideas if there are any possibilities of a model that are being that could be trained, which I am kind of missing out, please just tell me. Like this kind of a model is possible, like so that the model takes this and does that. Just tell me, then I'll see what I can do with it. If there are any models that are useful, which could be useful and that I'm ignoring out. Maybe because I still don't know much about the cell itself. Yeah.
I'm going to create an issue, give them feedback on...
Just give me a rough idea of anything,
Alright, so we'll do that. We'll make an issue on this, or give my feedback, and this is the same way, sort of, as we did with Ujjwal. If you have ideas about feedback on, maybe some ideas that Mayukh might incorporate, or if you see, different...
what kind of a model would be useful again? Like if I'm missing something out. The models that I have been training, I consider them to be useful or practical. there might be something that I am missing out, right? Some models which are needed, but which are not there, but which are they're like the possibilities. I don't know all of the possibilities. Right. If someone has any ideas, then just tell me.
Well, I mean, yeah, what might be useful too is to create like a document with like a list of things that people, might have as, because you wouldn't necessarily be implementing them this summer, but it would be like, what would be good to have to incorporate.
Exactly. That's what I want to know. What would be good to have?
Yeah. So actually, there's in the code section here, there are some directories. I'll bring people's attention to them. We have definitions and concepts, and we have worm movement. I think definitions and concepts might have what I'm looking for. I have a document on meta features, which defines some things. They're very general, like things about these meta features that we were talking about. But I think maybe in this area we can, or maybe another directory here at the root of GSoC 2020, we can create a document where we can request like different models or discuss like different things that maybe we should incorporate into this. I mean, now that we have a good start on it, a good running start, we can see kind of what is going on. So I don't know. It could be machine learning models. It could be maybe other types of predictive models. And just keep in mind that they don't necessarily have to be implemented, but it's just something to think about. If it's something that's easy to implement, we might do it. But if it's something harder, then it's something to think about, and maybe they'll be implemented later. But let's go back to projects here. Okay, and then so I think that's all for Mayukh there. That's work on embryo cell movement data. That's what he's doing. It's in progress. For Ujjwal,
I think the task number 3 in to-do list is completed.
Task number 3. Oh yeah, it's in
to-do list.
Yeah.
The left.
Yeah. Oh. This one?
The task. Task number 3, just below.
This one here.
Open DevoWorm text background accessibility, create more contrast between the fonts and background.
Oh, I'm missing it.
In a to-do list. In the to-do list.
Oh, okay.
On the left-hand side. On number 3.
Number 33. Or
number 3. Number 32.
Oh, I'm not seeing
this one.
This one? Oh, okay, never mind.
Yeah.
Alright, that was it. I was missing it for some reason. Okay. Alright, so that's done?
Yeah.
Alright. Thank you. Yeah. And then
and the integration with Miro-board, there is no possible way to integrate Miro-board with GitHub, so I think it's not gonna work for us. We can delete this.
I think that was just something we discussed. Early on, and so we'll just update that and then put it in finished so that we have. Okay, good. And then we have create pages, number 17. Reorganize, I guess.
Oh yes, this is in to-do list, in progress list. I am working on DevoZoo and Devo method pages right now.
Changes in design scheme, number of clicks to destination reduced.
This is done, I guess. Now the design scheme and UML diagram is finalized, I guess.
Okay. All right, that's done. And then I think the weekly blog posts are something that you guys are doing. It's not required, but you can keep doing those if you're doing them and give updates. The optimized color palette is something I know that Ujjwal said he was working on in Slack, or he said in Slack that he was working on it.
Yes.
And I think that's it. So I think that's good. Keep up the good work.
But I'm curious that if...
Oh yeah, what was that? Okay. Keep up the good work. I sent the evaluations in this week, so hopefully you saw them, and it's pretty good. I mean, I'm pretty pleased with your performance, and I always have to give that one thing that's like the suggestion for next time, or for you know, that can be improved upon. And I think I mentioned in your, and I do this every year pretty much, I mentioned to keep you know, good documentation and that sort of thing. So I think that's a good skill to have. You know, you can never have too much of that skill to keep up with your documentation and things like that. I can't remember, I mean, I made another comment, but those are just meant for you to kind of draw your attention to things. If you're already doing them, that's good, but I wanted to make sure that I had, you know, there's the room for improvement, and to make sure that you're getting the most out of the program, and that we can, you know, a lot of the open source software, you know, relies upon not just people contributing to projects, but the documentation, so that when someone else wants to do something or use it, it's transparent. So I was gonna actually do 2 more things this meeting. I'm gonna go over some papers, and I'm gonna briefly first talk about the Neuromatch Academy, which is something that is going to happen soon. And my other group actually is pretty deep into this academy. So it's a summer school for people who want to learn about neuroscience. And I've created a study group for this. And I've done this in consultation with the larger summer schools. Let me put this in the window here. And I was working on this with Jesse, so thank you, Jesse, for all your assistance and our sort of collaboration on this summer school. Yeah. So basically what this is this is a summer school for people who want to learn about basically computational neuroscience. So it's neuroscience, but it's computationally oriented. And so this is an open school. There is an enrollment, a formal enrollment, but a lot of people, you know, don't really have time to enroll for a full summer school. So they're actually making some of their materials available online, and they have them on GitHub. So there's a GitHub Match Academy is the location of their repository, and they have a lot of different things in the repository. They have lessons. They're doing a lot of their activities in Colab notebooks. So it's, you know, it's something that people in the meeting here are fairly familiar with. There's some resources for, you know, kind of prepping yourself for so, you know, you have to come in, you have to know a little bit about programming, a little bit about math, linear algebra, things like that, and a little bit about neuroscience. And so the prep is mostly, you know, sort of discussing things, you know, concepts that you'll need to know to do successfully in the school. But the school is actually meant to be pretty blank slate. You don't need to know a lot about any of those topics to participate. And so, you know, for a lot of you in this group, you might not have time to do the summer school, and it's, you know, but there are actually some things in these repos that are, you know, something you can take something away from. So there are lessons on, you know, different topics in neuroscience, different topics. So this is the course outline. People, they have a series of lectures on models and model fitting. There's some machine learning, and then you get into statistics. So you get into linear systems, Bayesian statistics, a little bit of optimal control, which is like control theory, a little bit about reinforcement learning, and then getting into like neurons and neural networks and neuronal networks, which are real neurons making networks. And so, you know, it's a little taste of everything. So if you're interested in, you know, learning a little bit about neuroscience, you might get involved in this. We have some resources here that we've prepared in the course of the preparation for the summer school that are available on the web, and this is going to be the site where they're hosted. So there are a bunch of different tutorials. Some of these are actually from DevoWorm ML. I've recycled them. Some of them are from other groups. And so, you know, check it out if you have time, if you're interested in learning about neuroscience or about, I think for this group, computation, you know, maybe some of you are going to get something out of the computation part, but I just wanted to bring everyone's attention to that. So, that's it. If you're interested, contact me. I know Krishna asked me about learning about neuroscience, and I didn't have a good answer for him at the time. So it's, you know, it's not one of those things I can give you a 15-minute tutorial on, but this is actually a good substitute if you go through some of these materials, and we can talk more about like how to, you know, learn more about neuroscience in a way that's not where you don't have to take a full course or you know, don't get a superficial overview of it. So
yeah, I would just add there's still opportunity to join or at least do the observer track for it, I believe. So you can check out the lectures, and also there's a discussion forum on neurostars.org, which is sort of a general, will be more for the course specifically. And then definitely, if you are interested, like I'm going to be fairly involved with it, in the other lab group as well. So I mean, there's actually ways where you can participate through that, and the links that Bradley sent. So you can interact with some things on GitHub, or you know, there's a Slack and a Keybase. So there's a lot of ways to be involved still. And I encourage people to do that, either through just looking at the Neuromatch stuff itself, or linking up with what Bradley is kind of helping to facilitate elsewhere. And it's a really cool. I'm really impressed by the community behind Neuromatch. They did two impromptu conferences earlier, like Neuromatch 1 and 2 on conferences virtually, and led to the summer school. And it's a very new thing. It's kind of only been in existence for a couple months. So there's some hiccups, but it's a really great endeavor. So check it out if you can. That's my plug for it.
Yeah. Thank you, Josie. Yeah. So we can talk more about that offline on Slack. So the paper I wanted to talk about today was this is a new paper, and something I've talked about in the group maybe a little bit, but we haven't really done anything with it. It's called Topological Data Analysis. And so the reason I bring this paper up in this group is because it is actually something on morphometrics. And so this is really nice. This is a really nice introductory paper to topological data analysis, but also applying it to morphometrics and developmental morphometrics. So the abstract, I like this first sentence. Shape is data, and data is shape. And so we've been looking. Actually, the paper Ujjwal talked about, the 3D MMS, is a good kind of example of that, where you can take data and you can build a model of shape. And we've had a couple of papers like that where, you know, you want to build a model that's geometric, it has a shape, a volumetric model where it has a shape, and extruding that from some data that we're collecting. So we have this organism that's like a complex shape, or this embryo, you know, that has a certain shape, the cells and the actual embryo. We extract data from that so we can measure it, but then we have to reconstruct the shape on the other end. And so that's kind of what they're doing here, but they're going a step further and then saying, how can we produce meaningful parameters to describe those shapes? And so biologists are accustomed to thinking about how the shape of biomolecules, cells, tissues, and organisms arise from the effects of genetics, development, and the environment. Less often do we consider that data itself has shape and structure, or that it is possible to measure the shape of data and analyze it. So they want to know, like, how do you measure that shape and analyze it and say something statistically meaningful about it? Here we review applications of topological data analysis to biology in a way accessible to biologists and applied mathematicians alike. TDA uses principles from algebraic topology to comprehensively measure shape in data sets. And so they use a function that relates the similarity of data points to each other. We can monitor the evolution of a topological feature. So these are things like connected components, loops, and voids. And so topological features are those things in the shape that, they're interesting features of that. You can think of them as features or a feature space, but they're mathematical objects, basically. So you have loops, connected components. I didn't bring this GIF, but there's a GIF that shows, I think it's on the Wikipedia page for like topology or one of those topics where it's like they have a donut that morphs into a mug with a handle on it. That's kind of the idea behind topological analysis, where you have like this shape, it's like a donut, and then you transform it into another shape that might share some of the features, it might not share some of the features, but the description there mathematically is to characterize a transformation. So there's a lot of math being done in this area. So like a lot of the machine learning stuff, we're interested in like, segmenting images or building maybe a volumetric model. But there is a second part of that, which is the analysis of the shape itself. So that's the evolution of topological features. So this evolution is a topological signature. And so this concisely summarizes large complex data sets. So this is a TD, they call this a primer. So this is nice and accessible. They kind of do a lot of this. This is kind of a vision statement. They show how it can be applied. And so this is the introduction. They make the point that shape is foundational to biology. So indeed it is. If we know, their shape is very important, say in leaves or in embryos or in mature organisms. And I think we've talked a little bit in a couple weeks ago about Darcy Thompson, who did a lot of work about 100 years ago on analyzing different transformations in shape. And they're kind of taking off from that point. Actually, they do mention Darcy Thompson here. Beyond observation and documentation, we can measure shapes just as we analyze data. Darcy Thompson, in his On Growth and Form, uses shear mapping to linearly transform biological shapes between each other and described something called allometry, or the relative growth of parts in an organism to the whole. We haven't talked about allometry in the group, but I may give a lecture on it in a couple weeks. These are sort of linear scalings of, so if you have an organism and it has, say, a beak, and the beak changes shape, okay, with growth or with evolution, you can put landmarks on different parts of the beak and look at different beaks from across nature. So from across species or across development, and you can figure out, there's a growth trend that happens between those two landmarks. And so the growth trend is characterized as allometry. It's just a mathematical function that describes sort of the transformation of that metric. So, sometimes people will scale head size versus body size, and they'll get a ratio. And that ratio then scales as the organism grows in size, it will scale, maybe linearly or non-linearly with growth in the organism. So this is something that we can do as well and understand what's going on with phenotypes. But this is an example on the left of what they've done with a leaf here. So they've looked at this leaf and they're kind of decomposing it mathematically. They're building this this network, I guess, of things along the surface of the leaf. And of course, the leaf is basically a vascular network. But they're decomposing it in this way. And they're looking at the variation across different leaf types. So you can see there's variation in the leaf type. But they're basically decomposing it into maybe common features. So they're using this network to find common features across these leaves. Even though they look to the eye very different, you can use different types of analysis to get information out of this. And if you're interested, you can go through this article, and they mention a bunch of different analyses. They throw out like Prochrist's analysis and geometric morphometrics, those sorts of things. So those, that's something you can look up later. The reason, the way people write an article like this is they pack it with a lot of references and mentions of things, and then your job is to look up those things later. But they actually do then take these networks and find the principal components. So we've talked about principal component analysis, where you plot the one principal component versus the second principal component, and that describes a lot of the variation in the data set. So with regard to these sets of leaves, you get distinct groups on your in your principal component analysis. Another way, so then they talk about that, and they talk about different methods. Now they get into what they call TDA, or topological data analysis, and this is what they call middle ground between mathematics and biology. So TDA is they kind of go through TDA here, and I've never really fully understood what it is or how to do it, but I've always wanted to get into it. It's the tools are coming online. It's a relatively new technique. It's something that in the mathematics community, they've done topological analysis for years, and they're just kind of coming up with tools. But sometimes the tools aren't really suited for the data or the application, and so I think now it's sort of coming together so that people can use these in a good context. So they talk about, in a mathematical context, networks are referred to as graphs. Nodes or points are referred to as vertices, while links between nodes and edges. So that's like a network type of analysis. So they use graphs. We can generalize the idea of graphs by adding triangles that link edges, or even tetrahedrons that link triangles. More formally, we can think of our data as composed of different building blocks called simplices, and that's a mathematical term. Vertices, edges, and triangles are 0, 1, and 2-dimensional simplices, respectively. A collection of multiple simplices makes a simplical complex, or complex for short. So now you're building from networks to these simplices to these simplical complexes. And then this is how you proceed with topological data analysis. You can describe the topology of a complex based on the number of its connected components, loops, and voids. So this is an example here. This is a complex where you have one loop, which is this, and one void, which is this triangle, 3-dimensional triangle, or this pyramid, and then they're connected by this connected component. So you have, these are the connected components, but this is the connection here between them. And there's a similar concept in graph theory, but in this case, they're just talking about basically creating a data, or a network out of the data, and then, analyzing it like a network or like a mathematical object. So they talk about the Vietoris-Rips complex, which is something very technical. So the VR complex starts with data in a metric space in a fixed non-negative parameter R, which is the radius. If the two vertices are close enough, that is, the distance between them is less than r, then the VR complex will have an edge between those two vertices. So this is a criterion for forming edges from a bunch of nodes. And there are other ways you can actually do this in graph theory. People will have done this in different ways, but this is the way they're suggesting that it be done in TDA. So they walk through an example of how to apply this to a data set. So you have these points in your, like, in our case, we have points that are the center of a centroid of a biological cell in an embryo, and you would take those points and you would join them. And so we actually wrote a paper on embryo networks, and this is a similar thing that we've done already in the lab. But now you can take those networks and you can find shapes within them. And so then they take these VR complexes and they create persistence barcodes, which are these structures here that kind of demonstrate like the distance from the the distance of the radius and different objects, and they create this visualization here that describes it. And then representing persistent features. So all observations described above can be summarized using two topological features: connected components and holes. And so connected components are the things that are connected like a graph, and holes are the things that aren't. And again, that has a parallel with network theory, where they talk about connected components. That goes back to Renyi, who's is a mathematician from a long time ago, and he's described connected components as something that was a feature of large networks. And so you can use a similar concept here, whether parts of the network are connected or not, to describe its structure. And so we can also visualize the connected components. And I'm not going to walk through it much more, but I just wanted to give people a taste of what this is about. And so now you get into applied topology. So this is the topological analysis, where you have the different shapes. You can transform between two different shapes, or understand the sort of the features in a shape. And I think this is actually quite useful to machine learning as well, because we're talking about feature selection and feature discovery, and this is kind of what they're doing here. But they're doing it in a little bit maybe more rigorous way for three-dimensional objects. There's more of a mathematical bent, so there's more formality to it. And so this is an application of TDA to biology. They have a nice figure here. So structural biology, which is A. B is evolution, so they're looking at, I guess, the genetic distance between samples. Letter C is an application of cellular architecture. So this looks rather familiar to the people who are segmenting cells. D is branching architectures. So this is like for leaves, or for like the descent of different species and genetic lineages. You can model them as a tree, and then understand the tree better. And then this E is a mapper. So this is where you're mapping across different points. So this is a hand, and they're analyzing this hand as a network. So they're mapping the hand to some, directed network here. So, and then they go through all these applications, and they've got some really nice figures in here. So, there's so many different techniques in here. I don't want to even get into it much more, because I really couldn't explain it in an hour, probably what they all are, or probably ever for some of them. But they have some nice figures in here. And it's good if you're interested in this more. We can talk about it. I think it's a very interesting approach, and it might be useful to the machine learning people, or even the biology people, for doing some analysis of development. And so Yeah, and then they end this paper with a great figure here. Endless forms of most beautiful X-ray computed tomography scans of the biological specimen showing the diversity of morphology in the natural world. So these are all these images from different plants, from seeds and other parts of the plant. This is a pepper, bell pepper. You know, you have all these. So this is what the kind of data you can collect. And we actually talked. I think it was Krishna who wants to build this general model. So this is actually quite relevant to that as well. We have all these different forms that we can maybe unify into a single model. And so this might be good for Krishna to read to get a sense of like what people are thinking in terms of going very far afield and doing all these different, you know, analyzing all these different types of forms and the types of data we might be able to get. So I think CT scans are a good way to get those data, but I think that that's not necessarily something that's publicly shared too much, but we can investigate that. So that's that paper, and again, I recommend that you look it over. Let me see if I can give you the link to the directory. I'll post it later for people. Okay, so yeah, Ujjwal asked, these papers are very interesting. Can you please share the link? There's the link. Jesse says, I'll have to show my Darcy Thompson meme about Cookie Monster. Yeah, it's in the Slack, so look at it. Cool, it comes up again here. Also, lots of thoughts with regards to affordances and direct perception. Yeah, we did talk about that link between morphology or morphometrics and direct perception and affordances. We should talk more about that. Yeah, I'll have to show this to Anson.
Yeah, I first put up Anson, and a lot of his things kind of fit with that. And then this isn't the place to go into my investigation of direct perception and affordance, but it's just, I was like, the first sentence that you pointed out in the abstract, it's like, oh, that, you know, comes up here again, too. And I'm really curious along the lines of what you said about how, what's it called, TDA? Is coming along. Like I'm curious. I have no idea. I've never heard of it before in my life, but I'm going to be curious to look into it regarding a lot of the other projects and stuff that we mentioned. So definitely interested in that.
Yeah, I think like it's like that type of analysis, topological analysis, hasn't really necessarily been accessible to non-mathematicians. But I think they're creating tools now that are coming along in terms of opening it up to other groups of people. You know, probably still want to talk to a mathematician about like how to do it correctly and get good results. But I mean, that would be something that definitely it opens up new avenues for a lot of things. And so, I mean, these are outside of biology. You could apply it to perception, I suppose, as well. So yeah. Well, thanks for attending, everyone. Yeah. We're at the top of the hour, a little bit past. Next week, we'll have, again, updates from Ujjwal and Mayukh. We'll talk about maybe the integration of the models into the Open DevoCell. And if anyone has anything else they'd like to present on or talk about, let me know. We can put it on the schedule. Otherwise, thanks for showing up, and talk to you guys next week.
Okay,